All right, it looks like we're ready to begin. So let's get started. Hello again, and welcome to Biocellugen's webinar series. It's great to have you all with us today. So in today's session, we will be focusing on two leading drug modalities. They are the T-cell engagers or TCEs and the CAR T-cell therapies. Before we dive in, let me start with a quick introduction. My name is Yuan Tian, and I am the marketing manager at Biocellugen Boston Corporation. I'll be moderating today's webinar. And I'm also very pleased to be joined by two outstanding speakers today. They're Dr. Daniel Powell and Dr. Baihong Liu. So first, it's my honor to introduce Dr. Daniel Powell. So Dr. Daniel Powell is a professor in the Department of Pathology and Laboratory Medicine and the Department of Obstetrics and Gynecology at the University of Pennsylvania. Dr. Powell is an international expert in cancer immunobiology and translational immunotherapy. He studies the role of the T-cell response in the control of human cancer, focusing on ovarian cancer and the application of adoptive lymphocyte immunotherapy, immunomodulation, and cancer vaccination. Dr. Powell is also the scientific director of immunotherapy in the Division of Gynecologic Oncology and is actively developing clinical trials of CAR T-cell therapy for ovarian cancer. And prior to being recruited to UPenn, he developed combinatorial tumor vaccination approaches, investigated the role of immunosuppressive regulatory T cells in cancer, applied adoptive TIL therapy for the treatment of patients with metastatic melanoma, and defined the factors associated with effective adoptive T cell therapy at the National Cancer Institute. Dr. Powell is well recognized for a wide array of contributions to the field of cancer immunotherapy. He is immediate past chair of the NIH CIC studied section, holds multiple patents on cell and gene therapy, serves on various federal academic and industry committees, and is a recipient of various awards, including the Outstanding New Investigator Award by the American Society of Gene and Cell Therapy. So we are truly honored to have you with us today, Dr. Powell. Thank you so much for joining us. And now it's my pleasure to introduce our second speaker, Dr. Baihong Liu. So Dr. Liu earned his PhD in bioengineering from Jiangnan University in 2015. He joined Biocellugen in 2017 and has since focused on antibody discovery and early stage research and the development of bispecific antibody therapeutics. And over the years, Dr. Liu has led and contributed to multiple bispecific antibody programs at Biocellugen, and several of which have successfully advanced to the preclinical candidate stage and into clinical development. Thank you so much for being here today, Dr. Liu. We are really looking forward to your presentation and your insights. So now you've met our speakers. Here's a quick overview of today's agenda. We will begin with Dr. Powell, who will share recent strategies in next generation TCE and CAR-T development. And then Dr. Liu will follow with a discussion on solid tumor TCE development and introduce Biocellugen's novel TCE design that integrates the VHH nanobodies. So with that, before we get started, just a quick note, today's webinar is being recorded and we will have a live Q&A section at the very end. So as you're listening, feel free to submit your questions at any time using the Q&A box. We will do our best to get through as many questions as we can after the presentations. All right, I think now we are ready for the presentations. Without further delay, I'll hand things over to Dr. Daniel Powell.
Wonderful. Yuan, thank you so much for that wonderful introduction. I'm curious, can you see my slides?
Yeah, we can see your slides.
Wonderful. Well, then I will get started. So greetings from the University of Pennsylvania in Philadelphia, Pennsylvania. I'm here at the Center for Cellular Immunotherapies, where we have more than 30 faculty focusing on the cure of cancer, both solid and hematologic malignancies, largely focused on the application of adoptive cell therapy. And so the vast majority of my presentation today will be focused on adoptive cellular therapy, but we also are working in the area of bispecifics, also known as T-cell engagers, and as indicated, CAR T cells. And so what I'd like to do for today's presentation is focus on a rather new application in terms of both CARs and T-cell engagers, and that is through the development of next generation single chain FV fragments that allow us to look at intracellular protein targeting. So with that, I will begin. I think it's worthwhile as we consider this type of application that the vast majority of therapies that are approved are focusing on cell surface antigens, and fewer are focused on targeting intracellular antigens such as neoantigens. We understand that tumor-infiltrating lymphocyte therapy can be very effective in the context of metastatic melanoma, where neoantigens or mutated non-self antigens are readily available to be targeted, and that can lead to curative therapy for a subset of patients. There's great excitement about targeting neoantigens or mutated self antigens. As indicated here, the broad neoantigen targeted therapies market was valued at nearly $2 billion in 2024 and is expected to grow to about $3 billion by 2030. So TIL therapy has a place and certainly has been effective for those patients with metastatic melanoma. But in order to bring this to the masses, we've looked at synthetic biology to advance the targeting of neoantigens. This has come in the form mostly of TCR-based therapy. So the collection or isolation of a T cell receptor that can then be transferred to a non-reactive T cell and allow that T cell now to see the intracellular protein. And a great deal of work has been conducted in this area. Of course, we do have a recent FDA approval of TCR-based therapy for synovial sarcoma. The advantage of this type of approach, again, is that the TCRs allow the T cell to see inside of the cell to target those intracellular antigens that are processed and presented to the immune system, presented by HLA or MHC molecules on the cell surface. And these can be highly selective with regard to their binding to these neoantigens in the context of a certain HLA allele. Some of the disadvantages of that approach is that the TCRs can be low affinity depending upon how they are isolated and where they are isolated from. In some cases, they can be promiscuous with regard to their specificity. And oftentimes, when targeting class 1 epitopes, CD8 co-receptor is required to increase the overall avidity and activity of the cell. This gives way to CAR-T cells, a completely different approach, which harnesses single-chain FVs to redirect the specificity of the T cell. Here, the single-chain FV on the cell surface allows these antibodies then to bind to cell surface antigens to trigger activity inside of the cell and mediate cancer regression. We now know that this is a very effective approach in certain hematologic malignancies with multiple FDA approvals. Some of the challenges that exist here are the possible on-target toxicity, which could limit its availability for some patients and limit the targeting of certain antigens. And CAR-T cells, unlike TCR-bearing T cells, require very high levels of antigen on the cell surface for recognition. So they cannot recognize very low copy numbers, and because they recognize cell surface antigens, they do not generally recognize antigens that are processed from within the cell. And so at this point in time, the field has really focused on TCR therapy to target those intracellular antigens. But we questioned whether or not we could change this way of thinking. Is it possible that we could create CAR-T cells that have the capacity to see inside of the cancer cell and to recognize those intracellular proteins now presented in the context of an HLA molecule on the cell surface? As we began to investigate antigens to target, we focused in on what is the most common mutation in all of human cancer, and that is mutations in KRAS. This oncogenic event occurs with high incidence in pancreatic cancer, in colorectal cancer, and non-small cell lung carcinoma, and in uterine cancers. For immunologists and immunotherapists focusing on targeting those mutated antigens, there's great interest in targeting the G12 loci of this molecule, as 80% of the mutations occur in that particular locus. Keeping in mind that when an antigen is presented to the immune system, it is presented by HLA molecules. There's a wide array of alleles. Depending upon your origin, you're more likely to have, for example, in Southeast Asia, a HLA A1101 allele. In the US, you're more likely to have an HLA A0201. So keeping this in mind, our focus has been to develop reagents, specifically single-chain FEs, that would allow us then to focus in on mutated KRAS antigens that are presented in the context of the HLA A11 allele. We began through collaboration with Beatrice Carino and Jerry Lynette at the University of Pennsylvania. In their work, they had performed mass spec to look at cell surface antigens derived from KRAS presented in the context of different HLA alleles. What you see on the left-hand side of this slide is the results of that mass spec analysis. You can see in the red that multiple alleles can present mutated antigens, mutated KRAS antigens from that G12 loci. These include A0301, A1101, and B702. And one can quantify the amount of antigen that's available on the cell surface. For example, in this cell line here, the Coral 23 cell line, in the context of A11, you can see the number of copies that are available. Well, working with Beatrice and with Jerry, they generated a number of T-cell receptors specific for these antigens, in particular focusing on the G12V mutation, and applied those TCR engineered cells to cancer cell lines, a wide array that were engineered for expression of HLA A11 so that the antigen could be presented. And they evaluated the capacity of those TCR transduced cells to recognize and to kill these cancer cell lines. And you can see across a wide array of cancers that immunologically, that G12V antigen is available on the cell surface, that these can be seen by the TCR transduced T cells. And in this 4-hour lysis assay, they can mediate the elimination of those cancer cells across a wide array of E to T ratios. So the takeaways from this earlier study is that select mutated GRAS G12 epitopes are presented. They're presented both in the context of A3, A11, and B7 alleles. We did not find the presence of G12C epitope on the cell surface. That's not to say it's not there, but at the level of sensitivity, we could not detect it. And we did not find those antigens presented in the context of HLA A2. Again, this may be an issue of sensitivity of the assay, but G12V is presented. It's presented and it's abundant in the context of HLA-A11 cancer cells at a low but immunologically detectable level. So keeping this in mind, we now have a good target antigen that has been validated for its expression and its targetability. With this in mind, we proceeded to try to isolate single-chain FVs that were capable of recognizing the mutated antigen in the context of HLA-A11. And to do this work, we teamed up with a private Australian company called Mirio. Mirio has a retained display system that allows one to generate single-chain FVs against peptide MHCs. They do this through the deployment of lambdoid bacteriophage display libraries. On the head of the bacteriophage, there's multivalent presentation of the single-chain FVs. They utilize paramagnetic beads that are coated with numerous peptide MHCs so that you have high avidity interactions. Important to this process, their library has lossless panning. So you continue to have diversity in the single-chain FVs. as you continue to pan for single chain FVs that recognize specific peptides in the context of HLA. These can then be made into soluble antigens that, the peptide MHC can be made into soluble units with fluors that are bound or conjugated, and that can then be used to detect these single chain FVs on the wall of bacterial cells for selection. And in so doing, one can then isolate a single chain FV that has the capacity to very specifically recognize a peptide in the context of a given HLA allele. So with this in mind, 4 single chain FVs were isolated. They are shown here across the top. I will refer to them as single chain FVs 2, 75, 7, and 14. What you can appreciate is when looking at its binding affinity, that the KDs are in this very low range of about 3 to 22 nanomolar. So good overall binding affinity. We then looked at the specificity of these single chains by testing them against 96 unrelated HLA-A11 peptide MHC complexes where the peptides are known to be expressed in vivo. And you can see that there was a high fidelity shown here in red for binding to the G12V peptide and not other peptides, even in the context of HLA. So no binding to the HLA-A11 molecule alone. It is highly dependent upon the peptide being presented. We then performed X-scan positional scanning. Here, this is kinetic tracing analysis that allows us to look at the binding to both the wild type peptide and the G12. The takeaway from this analysis is that the single chain FVs have a very strong preference for amino acids 5 through 9 of the peptide, the nonomer, for all of these single chain FVs. We then went on to perform an analysis of whether or not there was sequence-related off-target binding by viewing the immunopeptide algorithms, which predicted molecules or peptides that had similarity to the G12V epitope. We then pulsed them onto A11 cell lines and looked for relative binding, and you can see again, each of these single chain FVs was very specific for that G12V epitope in the context of A11. We then went on to perform targeting. And in order to do so, we began by creating bispecific T-cell engagers, which we're going to hear more about later in this presentation series. Here, what we seek to do is to create an immunologic bridge between the T-cell receptor from a non-engineered T-cell to a peptide MHC molecule that's presented on the surface of the cancer cell. And what you can see here is the results from, again, a 4-hour assay, a chromium release assay, looking at the amount of cell death that occurs when utilizing those TCEs or bispecific antibodies in cultures that have both the cancer cell and non-engineered T cells from donors. And you can appreciate in this particular assay, we have very good specificity. These cells here are K562 cells that are pulsed with the G12 epitope, or shown in blue, the wild type. So again, no specificity for the wild type antigen. The single chain FV redirects the T cells against the mutated antigen. What's remarkable about this technology is that it's able to distinguish a single amino acid difference between the mutated peptide and the wild type natural peptide. So with this in mind, having shown that T-cell engagers could be effective, we move forward with the creation of CAR-T cells. And here what we have shown in red across the top are the 4 different now CARs that are generated based upon the sequences derived from those single chain FVs. Looking down below, you can see this is the A2 binder is a non-specific CAR control. It's expressed well on the cell surface, and all of the other CARs are expressed well on the cell surface. We then looked for activity of those CAR T cells, specifically looking at their ability to bind to peptide MHC. And what you can appreciate is that for 14, 17, and 75, we have good binding to HLA A11 G12V dextramers, which easily detect that single chain on the cell surface of the CAR engineered cells. But interestingly enough, the O2 single chain FB was not capable of binding to the G12V A11 dextramer, suggesting that this is a low avidity or perhaps low affinity interaction. And I'll show you more data on that to confirm that this particular structure is unable to bind at high cell avidity to its intended target. And so again, we've generated CAR T cells. These CAR T cells rely upon the mutant KRAS single chain FB. They have 4-1BB co-stimulatory signaling along with CD3 zeta to allow them to now see target. We began by looking at interferon gamma responses against peptide pulsed target cells, and you can see that untransduced cells do not recognize either the wild type nor the G12V mutated peptide. The control CAR has no activity as well. 75, 7, and 14 all have very good activity against peptide pulse targets, even at very low concentration. However, the O2 variant of the CAR, again, which did not bind to the dextramer, had very low activity against even peptide pulsed target cells at very high concentration. So to further look at whether or not cell avidity was an issue here for the O2 CAR, we teamed up with Lumix, who has the Z-Movie instrumentation that allows one to look at cell avidity interactions. What we found was that when targeting a cancer cell line, Coral23, which naturally expresses the G12V variant and is engineered for A11, that the 14 CAR, which has very good activity against peptide pulse targets, bound to those cancer cells very well, but the O2 variant had no capacity to bind, at least not in the appreciable level. And so it appears that the relative avidity of these CAR T cells, the O2 CAR T cells, is not sufficient to drive activity. Well, we then went on to look at killing of the Coral 23 cell line with A11. And what we found was that there was very good killing by these 3 CARs that, again, all target G12V A11. You'll appreciate as we go through this analysis that you'll see that the 14 single chain FB outperforms the others somewhat mildly, but reproducibly, appearing to have the best overall activity. Importantly, when we don't have A11, we don't have killing. And so this is a both peptide and HLA dependent process required for the cell killing. With this in mind, we then moved to target human lung cancer xenografts in NSG mice. And so for these studies, NSG mice were inoculated with 250,000 of the Coral cell line. Again, this expresses the G12V epitope as well as the HLA-A11 allele. We then allowed it to grow for 7 days. This is an IV infusion, as this is a lung cancer model. Followed it by 2.5 million CAR T cells targeting G12V. Perhaps as expected, the O2 CAR had virtually no activity compared to the control CAR. This is aligned with the cell avidity results that we saw earlier. Alternatively, the 75, the 7, and the 14 CARs all had activity against the Coral A11 cell line. And you can see that there's a wide range of types of activity that are seen. And in particular for 14, there was deep regressions of some cancers, but not comprehensive for cancers in other mice. And so keeping this in mind, we were pleased to see that there was selective killing of the cancer cells, but because of the incomplete nature of these responses in some of the mice, we went back to the drawing board to determine how we could invigorate an improved response. And so to do so, we relied upon interleukin-12. It's a pluripotent cytokine. It's been utilized clinically. However, it's been toxic in patients with IV administration. Both IV and subcutaneous routes of delivery have been tested, but in all cases, there's been adverse events. So we questioned whether or not we could harness the activity of interleukin-12 by engineering it not only into the CAR-T cell, but have it expressed at a time when the CAR receives its signal through interaction with its intended target. So an activation-dependent release of interleukin-12. We tested this in vitro. When we activate the cells, there's release of interleukin-12. 2 days later, if you wash out the media, remove the stimulus, there's no longer any release of IL-12, so this is very tightly regulated. If you re-stimulate the cells, again, you get IL-12 release. And so keeping this in mind, we move forward with testing this type of approach to invigorate the KRAS CAR-T cell activity, keeping in mind that IL-12 has this benefit of not only reinforcing a Th1 response, but that Th1 response leads to T cells producing and secreting high levels of the interferon gamma. And interferon gamma can lead to an increase in HLA expression by cancer cells. And that's one of the limitations of targeting peptide MHC is the availability of peptide MHC or peptide HLA on the cell surface. And so this is a possible positive feed-forward mechanism to improve activity. And so we engineered the CAR T cells to have an activation-dependent release of human interleukin-12, and we tested the impact of this. When we have IL-12 on board, as I indicated earlier, we see improved or heightened interferon gamma responses by the CAR T cells. And if we take that media that has high levels of interferon gamma, we lay it upon cancer cells, we see an upregulation in the level of HLA molecules, indicating that they might be sensitized then to CAR T cell attack. In fact, that's what we see. So looking at spheroid assays, when we have interleukin-12 introduced into the CAR T cells, we have better overall killing, both against the Coral 23 line as well as YAPC, which has a lower abundance of the G12 epitope. And then we move that back into the very same xenograft model. Here, same number of cancer cells are administered IV, the same number of CAR T cells are administered now with interleukin-12 being released in an antigen-dependent manner. And what you can see is that, shown here in the red, that there's a increased efficacy of this approach. In fact, all of these mice cure their disease. Now, this provides a lot of rationale for advancing with this type of approach. However, you can also see with regard to the survival curve that although they cleared the cancer, all these mice died. And this is because of a profound GVHD that's mediated by the transferred human cells in the mouse. Well, we thought deeply about how we can overcome this as a barrier to make this a safe, effective approach. In order to do that, we have excised or ablated the endogenous TCR from those T cells that are engineered to have the KRAS CAR and the inducible IL-12. And when we do that, the T cell can no longer recognize the xenoantigens from the mouse. And because of this should be a very safe form of therapy. And in fact, that's what we see. So when we move back to the very same mouse model, we now see that there's a very profound regression of cancer. As we saw earlier, if you have IL-12, but you do not eliminate the endogenous TCR, you have toxicity. But if you now have a KRAS CAR with inducible IL-12 without its endogenous TCR, you now have therapy that leads to 100% survival of the mice. Interestingly, because you get resolution of disease, you find that T cell counts between day 18 and 25, they begin to drop. So once you have curative therapy, they're no longer required, their numbers diminish. We see a drop in levels of serum IL-12 to very low, negligible levels by day 25. And we don't find high levels of interferon gamma in the circulation of those mice. So eliminating the TCR does remarkable things to make this a safe yet effective form of therapy. So just to finish up, one question exists is, well, how does this platform compare to the TCRs that have been published upon? And so we reach back into patents. This is a patent that comes from the Far East for a KRAS TCR that targets the same antigen with the same HLA. And you can see in direct comparison that we have comparable, if not more favorable, activity in terms of killing of the coral cell line by the IL-12 KRAS CARs compared to the TCR. We also see improvements in interferon gamma responses as one might anticipate. And so we believe that this is a strong candidate to advance to the clinic. I will point out that we are working to improve the affinity of the single chain Fvs. For example, the 75 CAR and single chain Fv that we've studied throughout this presentation, you can see it has a modest overall killing capacity as a T cell engager or a bispecific antigen relative to new and improved high affinity single chain Fv clones that are recognizing, again, the very same antigen, and that is G12V in the context of A11. And so this provides an opportunity, perhaps even to eliminate the need for interleukin 12 activation dependent secretion. All right, so keeping all of this in mind, what I've shown you is that single chain Fvs or antibodies can be designed to target a single amino acid mutation in KRAS, distinguishing it from the wild type epitope, that these can be generated with high affinity and specificity that we see both in vitro and in vivo. I've shown you that these neoantigen specific CARs exhibit anti-tumor responses both in the dish and in the mouse, that armoring the CAR T cells with inducible IL-12 can sensitize the tumor cells to enhance killing by increasing their level of HLA and antigen on the cell surface. And by knocking out the endogenous TCR, one can improve the safety of this approach, thus widening the therapeutic index of this particular application. So with all of this in mind, we're now at a point where we are incorporating a new company called Katana Therapeutics. This is founded by 3 teams, ourselves here at the University of Pennsylvania, investigators at NYU, including Mark Yamarkovich, and Mirio Therapeutics. I focused on the application here of generating highly specific antibodies against mutated KRAS in the context of HLA-11. However, we have also performed similar analysis focusing on epitopes from PRAME in the context of HLA-0201. And so what we're bringing forward here is a wide array, a cadre of peptide HLA targeted immunotherapies, first beginning with targeting mutant KRAS in A11, and then bringing forward both CARs and T cell engagers focusing on PRAME antigens. So with that, I'd like to thank everybody for listening to the presentation, and for all the investigators who conducted this work. I want to point out Alex Benton and Jigang Liu, who really spearheaded this application of CAR T cell development. My collaborators at Mirio as well, and collaborators here at the University of Pennsylvania have made all this work possible.
Thank you so much, Dr. Powell, for that excellent presentation. That was really insightful. And we really appreciate you sharing your perspective with us today.
Thank you.
Yeah, while Dr. Liu is getting his slides set up, I just wanted to remind everyone that you're welcome to start submitting any questions you have related to Dr. Powell's talk using the Q&A box. We will be addressing those shortly, so feel free to drop them in as they come up. All right, I'll turn things over to Dr. Baihong Liu.
Okay, can you see my screen?
Yes, we can see it.
Cool. Okay. Good. Hello, everyone. I'm Baihong Liu from Biostrategy Beijing side. It's great to be here to give an introduction of our T-cell engager asset. Firstly, I will briefly talk about the challenges of T-cell engager in solid tumor and the recent innovations in the T-cell engager development. T-cell engagers have proven to be a promising class of cancer therapy over the past decades. There are 9 candidates have been proved until now, especially for the DL3 targeting T-cell engager, which make a big progress in the field of solid tumor. While for the conventional T-cell engagers still face some challenges such as T-cell exhaustion and the cytokine release syndrome. So the T-cell engagers still need to evolve by improving the efficacy and reducing the adverse effects. Actually, T-cell engager and ADC are 2 similar types of modality, both comprising anti-tumor antigen part and another part to perform toxicity activity. ADC generally has good controllability. We have clear understanding the toxicity profile of different toxins. Can better regulate the drug-to-antibody ratio when conjunction, and also can better control the drug administered to the human body. However, for the T-cell engager, it can trigger cascade amplification activation in the immune system. It's just like igniting a fire in a forest. It's difficult to control the extent of the burn. Recent reports suggest that reducing CD3 affinity can simulate low level of cytokine while maintaining on-target tumor killing. So the cytokine release and cytotoxic activity can be decoupled. Here, 2 studies demonstrated that when lower cytokine level in vitro, still maintaining tumor killing activity both in vitro and in vivo. Therefore, the attenuation of C3 affinity could be a strategy to improve the therapeutic index. But is it really simple to only reduce the C3 affinity? The answer may be no. Here we can see Amgen developed 2 PSMA CD3 T-cell engager with a high CD3 affinity version and a low CD3 affinity version. The high CD3 affinity version MG160 exhibited clinical activity, but also with higher, almost 25% of grade 3 CRS in the patient. For the low CD3 affinity version AMG 340, although exhibited only 2% of grade 3 CRS at even higher dose, the overall response was 0. In fact, the patient with solid tumor, only a small fraction of antibody can penetrate the tumor microenvironment, where have small number of T cell numbers and typically exhausted. Consequently, the low CD3 affinity version may not effectively trigger this T cell function. To address this challenge, some methods such as masking strategy are currently under preclinical and clinical investigation. Such technology can enable targeted T cell activation within the tumor microenvironment and minimizing systemic toxicity. Here we can see these two candidates by using masking strategy demonstrated promising efficacy signal in clinical study, and also with a well-tolerated safety profile. But if you don't want using masking strategy or don't believe this technology, and also want to keep the high CD3 affinity, I think we should find antigen with similar characteristic to the DL3. DL3 was proven to be a very suitable antigen for the T cell engager development because it's minimal expressed in normal tissue and highly expressed in the tumor. On the other hand, to address the T cell exhaustion issue, the tri-specific antibody with co-stimulator could be the next wave for the T cell engager development. This concept was supported by the second generation of CAR-T therapies, which combining co-stimulatory signaling and retrieves more robust and durable anti-tumor response. There are some tri-specific antibody with co-stimulator are under preclinical and clinical study. I hope they have good performance in the future. Now I will talk about our T cell engager asset. To address the T cell exhaustion issue, we developed DL3, CD3 from BB tri-specific antibody by integrating a 4-1BB antibody to further maximize the T cell function. This molecule was developed by using our fully human antibody discovery platform, which facilitates for the different format assembly. Then we can do a quick format screening in vitro and in vivo by using our biomass source pharmacology and the CMC platform for in vitro and in vivo study and the pharmacology and study. We test the different format of DL3, CD3 T cell engager, and then fuse the 4-1BB antibody to the different projection, and also test the different paratopes of 4-1BB antibody. After in vitro and in vivo format screening, finally we locked in this format. We applied a high affinity anti-DL3 antibody for the tumor location. For the anti-CD3 part, we introduced the steric hindrance to further reduce the CD3 binding avidity. As we can see on the right, the tri-specific antibody binding to the Jurkat cell was largely decreased compared to the bispecific antibody control and the benchmark. For the anti-4-1BB domain, we fused the anti-4-1BB with HH to further the co-stimulatory signal to enhance the T cell function. In the T cell proliferation assay, we can see our DL3 tri-specific antibody can more potent proliferation of T cell than the DL3 bispecific antibody counterpart under the benchmark. This data show that the anti-4-1BB domain can generate a synergistic effect with anti-CD3 domain. We then did the repeated stimulation IC. After 4 rounds of stimulation, we can see the PBMC treated with DL3 trispecific antibody can still keep the high cell viability and T cell cytotoxicity. However, for the DL3 bispecific counterpart and benchmark, they largely decreased the cell viability and cytotoxicity. This data demonstrated that the aid co-stimulator signal indeed enhanced T cell persistence and functionality. I think the most concern of CD3 from maybe trispecific antibody is whether it can induce the non-specific cytokine release. In vitro study by co-cultured PBMC and tumor cell line, we found our trispecific antibody can only induce the cytokine release in a DL3 dependent manner. But the DL3 trispecific control can induce the non-specific cytokine release in the absence of DL3 expansion. We then compared our DL3 trispecific antibody to a non-specific antibody control in our C3 and from-B humanized mice. After a full round of dose as high as 50 milligram per kilogram of our DL3 trispecific antibody, we can see in contrast to the non-specific antibody control, there's no bodily weight loss were observed in the trispecific antibody treatment group, and also no systemic cytokine release was observed. In vitro cytotoxicity assay, we can see our trispecific antibody can induce the more potent tumor killing activity. And our trispecific antibody can also effectively induce the cytotoxicity activity will lower the effector to target ratio and further upregulation of anti-apoptotic marker. We then evaluated anti-tumor activity in xenograft mouse model established from small cell lung cancer that expressed the DL3. We can see mice treated with a single dose of trispecific antibody can significantly inhibit the tumor growth and comparable to the benchmark. But the T cell proliferation in vivo was much better than the benchmark. Our trispecific antibody can also promote the in serum CXCR10 secretion. This molecule is now in a cell line development stage with expression level in the range of 3 to 5 grams per liter. Our trispecific antibody also shows good stability under stress conditions. Okay, finally, for the summary, we can see CD3 T-cell engager is a very complex system whose activity and the therapeutic window are determined by multiple factors. It's difficult to solve all the problems by improving a single condition, requiring a case-by-case study. Therefore, to address the T cell exhaustion issue, we developed DL3 CD3 from the trispecific antibody and demonstrated its potent therapeutic activity. Okay, that's all the content I presented today. Thank you for your listening.
Thank you so much, Dr. Liu, for that great presentation. Now I'll take it from here as we moved into the Q&A session. I saw that Dr. Powell has already answered a few questions in the Q&A box. And for those of you who haven't submitted a question yet, now is a great time. We will try to get through as many as we can. All right, let's start with our first question. I will start with the part for Dr. Powell. So the question about the modalities incorporated the novel KRAS construct. How do we choose from CAR-T, TCE, TCR, and TCE?
Such a great question, and it would be wonderful if we could have an active discussion with the persons asking the question. But your question is both timely and very important. The most simple answer is we don't know. So I'm familiar with data, and I'm going to be very careful because I don't want to make this constitute a disclosure. But there's instances where a particular antigen has been targeted as a CAR, as an ADC, and as a T cell engager in 3 independent trials by the same group. And in some cases, the platform dictates whether or not you have toxicity. Surprising to me was that in the context of this work, and please take this with a grain of salt because this is a personal communication, the ADC and the T cell engager were toxic while the CAR T cells were tolerable. So, I think we have to consider all the factors with all the knowledge that we have, but it's not a very simple question to answer. And I think there will be instances, particularly as we think of solid tumor, where one platform will not only outperform another, but also be more safe. Perhaps, when we think about hematologic malignancy, this might be more easily answered because the cancer cells themselves are more readily available, and toxicities are generally more limited than targeting solid tumor. But that's a wonderful question, and the field is deeply considering this. And if we could figure out a way to prioritize these platforms, again, to increase therapeutic index, high efficacy, and high tolerability, I think we would all be in a better place.
Thanks, Dr. Powell, for the detailed answer. And next up, I think there's an audience posted several questions. I guess I'll address them one by one. So the first one here is there evident IL-12 neo-CAR dual CAR better than combination of CAR and IL-12?
So we would never conduct that trial. So the history with administration of IL-12 as a soluble molecule is barren with regard to its results. So it's highly toxic. And so this is something that perhaps could be tested in a mouse model for direct comparison. But a CAR plus IL-12, and I'm assuming the question is related to systemic delivery of IL-12, would be anticipated to be highly toxic. And that's why, companies as well as academics like myself are working on approaches that localize the delivery of interleukin-12 so that you don't have the systemic toxicities.
Great. Thanks, Dr. Powell. And next question, does IL-12 function through boosting APC? Is other cytokine or factor considered for dual CAR?
Yeah, so really great question. We know that IL-12 has a profound effect on APCs. The model itself is not designed to test that hypothesis, but we would, based upon what we know of fundamental immunology, we would expect that we would get activation of APCs, hopefully locally in the tumor microenvironment that would enhance cross-presentation of antigens and invigorate an endogenous immune response to broaden the overall activity. And this gets into another question that I think was asked by this particular person, whether or not we could look at this in a syngeneic mouse model. And certainly that's something that could be done to evaluate the impact of APCs in that context. And then finally, with regard to the question of other cytokines, I think most of you may be aware of the experience here stemming from trials at the University of Pennsylvania that patients who previously received CD19 CAR-T cell therapy but had relapsed disease but still maintained a level of CD19 on their tumor blasts have now been administered CD19 CARs that release interleukin-18. Interleukin-18 is very similar to IL-12, also a pleomorphic cytokine. It engages and activates NK cells and CD8 T cells to participate in the anti-tumor response, probably more so for NK cells, as best we can tell. But that being said, other cytokines are being investigated. And in the context of the work that we're doing here, IL-18 has also been tested in preclinical models as well. So I think there's plenty of opportunity to extend this beyond interleukin-12.
Awesome.
I should also tell you that those patients, and forgive me for not pointing this out, those patients who went on to that trial of the CD19 CAR with IL-18, many of them respond. And off the top of my head, I don't recall the response rate, but I think it was more than 60% of patients, again, patients who previously received CAR T cells targeting CD19, now having a response with a secondary administration of CAR T cells with interleukin-18 being secreted.
Awesome. Thank you so much, Dr. Powell. I think that's also covered the third question raised by this person. So I'll move on to the next part for Dr. Baihong Liu. So the first question here is, have you conducted toxicity testing of your tri-specific antibodies in monkeys?
We recently completed toxicity testing in the cynomolgus monkey, and our tri-specific antibody demonstrated good tolerability at a dose of, I guess, 10 mpk. Okay.
Great, thanks. And next question here, why did you choose 41BB instead of the CD28 as the co-stimulatory factor for developing the tri-specific antibody?
Okay, that's a very good question. I think both targets are classic co-stimulatory molecules, each with a distinct expression profile and a functional feature. CD28 generally induced higher toxicity activity, and the 41BB can sustain the proliferation activity and the tumor killing activity. I think it's not feasible to pick a winner. It needs further clinical validation. So I think the selection should be tailored to the specific project requirements.
Thank you. I think we're getting close to the end of the session, so I'll take one question for Baihong and one question for Dr. Powell, and then we'll end the session. So this question for Baihong, what other antigens do your VHHs from the fully human nanobody mice target?
Yeah, we have successfully did antibodies discovery for almost 100 targets by using our fully human antibody mice, including tumor antigen targets and some IO targets. We will develop more targets in the future. For the detailed information, I think you can contact our BD. Thank you.
Awesome. Thanks, Baihong. And last question for today. Okay, so this is for Dr. Powell. What are your thoughts on extending neoantigen targeting technologies to self-antigens derived from overexpressed cancer-specific proteins?
Great. I see that question is from Rika. So Rika, that's a great question. And I didn't have time to go into this today, but yes, this is highly feasible. And we've already applied this. And so again, just in the last slide, I talk about another program targeting PRAME antigens. PRAME is an overexpressed cancer-specific protein. So it's non-mutated. And because it fits the mold of a cancer testis antigen, we can target it. Now, others, including Immatics, they've created TCRs against PRAME. And they've shown good clinical activity. I think more than 60% of patients show some level of response to TCR-based therapy targeting PRAME. So there's already, again, proof of concept for targeting this molecule. We already have single-chain Fv binders that bind with good affinity to the PRAME epitope in the context of HLA-A*02:01. So yes, it's not only feasible, it's been done. So we can extend the technology to target self-antigens. Good question.
Great. Thank you, Dr. Powell. Oh, sorry. I think that's all the time we have for questions today. So before we wrap up, I would like to extend a big thank you to Dr. Powell and Dr. Liu for sharing their expertise and insights today. And of course, thank you to everyone in the audience for joining us and for the great questions. If you have any follow-up questions, or if we haven't been able to answer your questions for today, feel free to reach out to us through our website at biocelgen.com or connect with us on LinkedIn. We will also provide follow-up answers on the LinkedIn page. So just keep an eye on our new posts there. We will be happy to continue the conversation. And in case you missed any part of today's session, this webinar will be available on demand, and you will be able to request a recording directly through our website. All right, I think that's all from us today. Thanks again for joining. Have a wonderful rest of your day, and we hope to see you at our next webinar. Thanks,
Thank you.
everyone.